for the human disease treatment or therapy, stem cell therapy. Some patients, if you inject the suitable stem cells, so these cells can regenerate the cells or organs in the patient's body, right? So that stem cell therapy is quite important in the biomedical or medical researches. So if we want to have the stem cells, for the therapy or for all kinds of research, right? The only way we have to have the fertilized eggs and to the medulla and to the blastocyst areas. There have the inner cell mass, and we have to isolate this inner cell mass from the blastocyst stage and to culture the stem cell. So the embryo is the only way to harvest stem cell. So you can imagine it's so difficult, and sometimes have the, if you want to cure the human's disease, you have to have the human's stem cell, right? So where to get these precious embryos? It's really a big question, right? So that's, people always think about how can we get more stem cells, not like a fully development, just like, pluripotent, that will be enough, okay? We, maybe we don't need to go to the fully development, can go to an individual, but we just can go to a neuron or go to other specific cell type. That's will enough for the therapy, right? So that's IPS. The people think about how can we harvest or how can we have another or alternative strategy to get the more stem cells for the study, right? So this Japanese scientist named Yamanaka, he's a very, very famous scientist in stem cell research field, and he find the four factors, four very important transcriptional factors. You have the somatic cell from your ear fibroblast. Then you transfer, you have four very important transcriptional factors into the cell, and this cell can be IPS. So that is generation of induced pluripotent stem cell lines. So that these cell lines can develop into neuron and also other cell types. So this is a great work. So he shared the 2012 Nobel Prize for the IPS, discovery of IPS. That is also from, from SCNT, from somatic cell to an individual, right? This is from somatic cell, you go to the stem cells for other types. That's a kind of, reprogramming of the somatic cells, right? That's the reason these two great scientists share the Nobel Prize. Okay, so today we will spend time for talk about the somatic cell nuclear transfer. That's techniques, several research programs in our country did a very excellent work. Actually, my group also can do the somatic cell nuclear transfer for the pig production. So here, it's kind of pretty easy if we know this somatic, if we know this principles. What this technology needs is only one somatic donor cell, right? With donor cell, we need to have the nuclear. And another is the unfertilized recipient egg. Unfertilized recipient egg. And electric shock. So electric shock, just once the sperm on all sides, fertilization, they need the energy to fuse, right?
Zinc spark.
Yeah,
There is a
so-
zinc spot.
Yeah, so here the like electric shock just mimic that energy burst to make the two cells fuse together. Like yeah, mimic the energy burst due to the fertilization. Okay, and surrogate mother you need to have a baby, right? So that's pretty easy. That's just a one donor cell, one unfertilized recipient egg, and the electric shock, and surrogate mother. So, so the challenge is how was Dolly created, right? If you cut yourself, new skin grows of the wound because the skin cells are programmed to produce new skin. So this is vital while you are healing or growing even means